Dear viewers, welcome to EPG Pathshala. I am Dr. Preeti Khandelwal. I work as Assistant Professor in Sri Venkateshwara College. The topic of my lecture is the cytoskeleton. In our today's lecture, we will highlight the following aspects. What is cytoskeleton? Components of cytoskeleton, structure of actin molecules and their dynamics, function of actin molecules, intermediate filaments and functions, structure of microtubules and their dynamics, motor proteins that are kinesin and dynein, function of microtubules, and in the end we will summarize the lecture. Let us begin with what is cytoskeleton. Cells require something to keep their shape intact, and the framework is provided by the cytoskeleton. The word cyto means cell. So, cytoskeleton is cell skeleton. Hence, the cytoskeleton may be defined as a network of fibers extending throughout the cytoplasm, which not only supports the plasma membrane and gives the cell an overall shape, but also helps in exact positioning of the organelles. Its primary functions are to organize the structures and activities of the cells. It helps in establishing cell shape. The cytoskeleton extends throughout the cell. It is attached to the plasma membrane and internal organelles, so producing a framework for cellular organization. It can be very dynamic and have components capable of reorganization in less than a minute, or it can be quite stable for several hours. Cytoskeleton also provide mechanical strength. It is responsible for cells movement, which includes not only the movement of the entire cell but also the internal transport of organelles, provide tracks for the transport of vesicles and other structure such as mitotic chromosomes through the cytoplasm. In eukaryotic organism, cytoskeleton is composed of three principal types of protein filaments: microfilaments, microtubules, intermediate filaments. Microfilaments are the polymers of the protein actin organized into functional bundles and the networks by actin binding proteins. Of the three types of protein filaments, the cytoskeleton microfilaments are the narrowest. Microfilaments are especially important in the organization of the plasma membrane, including surface structure such as microvilli. Microfilaments can function on their own or serve as tracks for ATP powered myosin motor proteins, which provide a contractile function as in the muscle, or ferry cargo along microfilaments. Microtubules are long tubes formed by the protein tubulin and organized by microtubule associated protein. They often extend throughout the cell, providing an organizational framework for associated organelles and structural support to cilia and flagella. They also make up the structure of the mitotic spindle, the machine for separating duplicated chromosomes at mitosis. Molecular motor called kinesin and dyneins transport cargo along microtubules and are also powered by ATP hydrolysis. Intermediate filaments are composed of multiple strands of fibrous proteins. Intermediate filaments are composed of multiple strands of fibrous proteins coiled together. As their name suggests, intermediate filaments have an average diameter of 8 to 10 nanometer in between that of microfilaments and microtubules. They are tissue specific filamentous structure providing a number of different functions, including structural support to the nuclear membrane, structural integrity to cell in tissue, and structural and better function in skin, hair and nails. Microfilaments. The major cytoskeleton protein of most cell is actin, which polymerizes to form actin filaments, which are thin, flexible fibers approximately 7 nanometer in diameter and up to several micrometers in length. Actin filaments are also called as microfilaments, which are organized into higher order structures forming bundles or three-dimensional networks with the property of semi-solid gels. The assembly and disassembly of actin filaments, their cross-linking into bundles and networks, and their association with their cell structure such as the plasma membrane are regulated by a variety of actin binding protein. Structure of actin molecules. Actin is the most abundant intracellular protein in most eukaryotic cells. In muscle cells, for example, actin comprises 10% by weight of the total cell protein. Even in non-muscle cells, actin makes up 1 to 5% of the cellular protein. The cytosolic concentration of actin in non-muscle cells ranges from 0.1 to 0.5 millimolar. In special structure such as microvilli, however, the local actin concentration can be 5 millimolar. The three-dimensional structure of both individual actin molecules and actin filaments were determined in 1990 by Kenneth Holmes, Wolfgang Kabsch and their colleagues. Individual actin molecules called globular or G-actin has tight binding sites that mediate head-to-tail interaction with two other actin monomers. So, actin monomers polymerize to form filaments called filamentous or F-actin. Each actin molecule contains a magnesium ion complexed with either ATP or ADP. The conformation of the molecule is affected whether the ATP or ADP is attached. Addition of cations induce the polymerization of G-actin monomers into F-actin filaments, and the process is reversible. Each monomer of G-actin is rotated by 166 degree in the filaments, which therefore have the appearance of a double-stranded helix. Because all the actin monomers are oriented in same direction, actin filaments have a distinct polarity and their ends called barbed or plus ends, and pointed or minus ends are distinguishable from one another. Addition of actin subunits is favored at the plus end, whereas minus end is favored for the dissociation. Dynamics of actin filaments. The in vitro polymerization of G-actin to form F-actin filaments can be monitored by viscometry, sedimentation, fluorescence spectroscopy or fluorescence microscopy. The mechanism of actin assembly has been studied extensively. The polymerization of pure G-actin in vitro proceeds in three sequential phases. The nucleation phase, which is marked by a lag period in which G-actin subunits combine into short, unstable oligomers, which reaches 3 subunits in length, acting as a stable seed or nucleus. The elongation phase, which is marked by a rapid increase in the filament length by the addition of actin monomers to both of its ends. As F-actin filaments grow, the concentration of G-actin monomers decreases until equilibrium is reached between filaments and the monomers. The steady state phase, in which G-actin monomers exchange with subunits at the filament end, but there is no net change in the total mass of the filaments. The critical concentration is the concentration of G-actin monomers in equilibrium with actin filaments. At monomer concentrations below the critical concentration, no polymerization will take place. It occurs only when the monomer concentration is above critical concentration and the filaments assemble until steady state is reached and the monomer concentration fails to critical concentration. Actin filaments, they grow faster at plus ends than at minus ends. The rate of addition of ATP G-actin is nearly 10 times faster than one end, the plus end. 10 times faster at the one end, the plus end, then at the minus end, the rate of addition is determined by the concentration of free ATP G-actin. It has been shown with the help of kinetic experiment that on average 12 subunits are added to the plus end every second, whereas only 1.3 will be added at the minus end every second. In contrast, the rates of dissociation of ATP G-actin subunits from the two ends are similar, about 1.4 per second from the plus end and 0.8 per second from the minus end. The rate of addition is dependent on the free ATP G-actin concentration, whereas the loss of subunits does not. The critical concentration for both the ends are different. At the steady state, the free ATP G-actin concentration is between the C plus or Cc for plus end and C minus Cc for minus end. So, the plus end will grow and the minus end will lose subunits. This process is called treadmilling. The mechanism of treadmilling is accelerated by two actin binding proteins, profilin and cofilin. Profilin is a small protein which binds G-actin on the opposite side of the ATP. Profilin is a small protein which binds G-actin on the opposite side of the ATP binding site. When profilin is bound to the G-actin, ADP is replaced by ATP, which yields a profilin ATP actin complex. This complex can bind to the plus end efficiently and profilin dissociate once actin molecule is incorporated into the filament. Cofilin is a small protein binds F-actin in which the subunits contain ADP that are present towards the minus end. It binds by interaction between the two actin monomers and induces a change which destabilizes the filament, breaks into short pieces. In this way, it generates free minus ends and the disassembly of the filament is enhanced. Assembly of actin filaments are controlled by a diverse group of actin binding proteins. The initial and rate limiting step in the assembly of the actin filament is the nucleation of actin monomers. The nucleation step is coordinated by two classes of proteins, formin and ARP2/3, actin related protein complex. Cells have another family of motor protein called myosin, which move along the actin filaments powered by ATP hydrolysis. Myosin 2 was the first myosin discovered from the skeletal muscle. Myosin consists of 2 heavy chains and 4 light chains. Tails of the heavy chain dimerizes to form a coiled coil and light chains are associated with neck region of the heavy chain. For the movement to occur along the actin filament, myosin head requires binding of ATP which is hydrolyzed to aid the movement. Myosin head has actin binding site also. Hence, the myosin head uses ATP to pull on an actin filament. There are about 40 myosin genes in the human genome, which implies that these genes are paralogous in nature. These genes are responsible for the type of myosin present in the cell, from which 3 types of myosins are most studied. These 3 classes are commonly found in animals, fungi, myosin 1, myosin 2 and myosin 5. The intermediate filaments. These filaments are only present in cells that display a multicellular organization. Interestingly, there are large number of different IF proteins, and not all cells contain the same type of IF protein. In fact, some cells contain no IF whatsoever. These proteins are divided into 4 major types based on their sequence and their tissue distribution. One type is keratins that are found in epithelial cells, hair and nails. Second is nuclear lamins, which form a meshwork that stabilizes the inner nuclear membrane. Third type is neurofilaments that strengthen the long axons of neuron. And fourth type is vimentins, which provide mechanical strength to the muscles and other cells. Intermediate filament associated protein, abbreviated as IFAPs, cross-link intermediate filaments with one another, forming a bundle or a network, and with other cell structure including the plasma membrane. Only few IFAPs have been identified, but many more will undoubtedly be discovered as researchers focus on attention on the proteins that control IF organization and assembly. Unlike actin binding proteins or microtubules associated proteins, none of the known IFAPs sever or cap intermediate filaments. Sequestered IF proteins in a soluble pool or act as a motor protein. Rather, IFAPs appear to play a role in organizing the IF cytoskeleton, integrating the IF cytoskeleton with both the microfilament and the microtubule cytoskeletons, and attaching the IF cytoskeleton to the nuclear membrane and plasma membrane, especially at the cell junction. An essential role of intermediate filament is to distribute tensile forces across cells in a tissue. The organization of intermediate filaments into networks and bundles mediated by various IFAPs provide structural stability to cells. Major degenerative diseases of skin, muscles and neurons are caused by disruption of IF cytoskeleton or its connection to other cell structures. Now, the third type of protein fiber, that is microtubules. Microtubules participate in a variety of cell activities. Most involve motion that is provided by protein motors that use ATP. They determine the positions of membrane enclosed organelles and direct intracellular transport. The migration of chromosomes during mitosis and meiosis takes place on microtubules that make up the spindle fibers. Structure of microtubules indicated that they are made up of 13 longitudinal repeating units, which are called as protofilament. Each protofilament consists of major protein tubulin and microtubule associated protein, which are abbreviated as MAPs. Tubulin is a dimer, consists of alpha and beta subunit. Each subunit of the tubulin dimer can bind one molecule of GTP. The GTP in the alpha subunit is non-hydrolyzable, whereas beta subunit is exchangeable with free GTP and hydrolysis. The protofilament have an intrinsic polarity as alpha subunit is present at one end and beta subunit at the other. The end which is favorable for the polymerization is the positive end, which is the exposed beta subunits, whereas the minus end has exposed alpha subunits. A single microtubule is built from the 13 protofilaments. In addition to the simple singlet structure, doublet microtubules are found in specialized structures such as cilia and flagella, and triplet microtubules are found in centrioles and basal bodies. All microtubules are nucleated from structures known as microtubule organizing centers or MTOCs. The minus end of the microtubule stays anchored in the MTOC. In interphase cells, the MTOC is known as the centrosome and located near the nucleus, producing a radial array of microtubules with their plus end toward the cell periphery. With their plus end towards the cell periphery. During cell division, reorganization of microtubules occur to form a bipolar spindle for the segregation of copies of the duplicated chromosome. In cilia and flagella, microtubules are assembled from an MTOC called a basal body. Centrosomes in animal cells consist of a pair of orthogonally arranged cylindrical centrioles surrounded by apparently amorphous material called pericentriolar material. Gamma tubulin ring complex present in the pericentriolar material is an important component of nucleating the cytoplasmic microtubule array. Let's talk about microtubule dynamics. Assembly of microtubules is catalyzed by the presence of microtubule associated proteins MAPs. The polymerization of microtubules involves three phases, which are slow nucleation phase, elongation phase, steady state phase. The assembly to occur, alpha beta tubulin concentration must be at the critical concentration. Tubulin dimers add faster to one end, which is the positive end with exposed beta tubulin. The critical concentration is lower at the positive end. The critical concentration is lower at the plus end than at the minus end. At steady state phase, alpha beta tubulin concentration is higher than the CC for the plus end, whereas for the minus end it is lower. This condition results in the net addition to plus end and loss from the minus end. This process is known as treadmilling. Microtubules also possess the ability to undergo dynamic instability, which is distinct from the phenomenon of treadmilling. Now, how the microtubule structure and dynamics are regulated. Microtubules are stabilized by binding of microtubule associated proteins MAPs, and among these proteins are tau family of proteins and tau itself, MAP2 and MAP4. The tau proteins act as spacer between the microtubules, and MAP2 forms fibrous cross bridge between microtubules and links them to intermediate filaments. Some of the MAPs associate with plus ends of microtubules, and in some cases only the plus ends of the growing, not shrinking ones. This class of proteins is called plus TIPs. Some of the TIPs stabilize the plus end against a catastrophe or enhance the frequency of rescues. Thus, promotes a continued growth of the microtubule, and others provide the aid for attachment to the organelle. Microtubules are disassembled also with the help of certain proteins such as Kinesin 13, family of proteins, and oncoprotein 18, OP18 or STAT-min. Kinesin 13 protein bind and curve the end of the protofilament into the GDP beta tubulin conformation, which facilitates the removal of terminal tubulin dimers, and the rate of catastrophes is increased OP18 is also binds to two tubulin dimers in a curved GTP beta tubulin like conformation, enhancing the rate of catastrophes. It may also work by enhancing the hydrolysis of GTP in the terminal tubulin dimer, and hence its dissociation is fastened. Next is microtubular motor proteins and movements. The major two motor proteins are responsible for powering the variety of movements, which are kinesin and dyneins. Kinesins and dyneins move along microtubules in opposite direction. Kinesins move toward the plus end, dynein move toward the minus end. Kinesin is a dimer of two heavy chains, each associated with a light chain. It comprises of the following domains: a pair of globular head domains binds microtubule and ATP, short flexible linker domain for forward motility, and a central stalk domain, dimerization of the two heavy chains, a pair of small globular tail domain that binds to receptor on the member of cargos. Kinesin is a highly processive motor protein. The double-headed molecules of kinesin takes 8 nanometer step from one beta tubulin subunit to the next on the same protofilament in the microtubule. This entails each individual head taking 16 nanometer and two heads work into highly coordinated manner along the microtubule. ADP is bound to each kinesin heads. Loss of ADP is induced when one head binds to the beta tubulin subunits, giving rise to the nucleotide free state and strong binding of that head to the microtubules. The leading head that binds ATP, which induces a conformational change causing the linker region to point forward, dock into the head domain, and so thus the trailing head forward. The leading head binds a microtubule and releases ADP, which induces the trailing head to hydrolyze ATP to ADP and inorganic phosphate. Inorganic phosphate is released and the trailing head can now dissociate from the microtubule and the cycle is repeated. Second motor protein is dynein. Dynein motor protein transport organelles towards the minus end of the microtubule. This is a two-headed molecule built around two identical heavy chains. A single dynein heavy chain consists of a stem and a round head domain containing the ATP's activity, from which protrudes a stalk having microtubule binding sites. Dynein requires the Actin, the protein complex which links dynein to the cargo and regulate its activity. Microtubule dynamics in mitosis. Mitosis is the process that separates newly replicated chromosomes equally into daughter cells. Specialized microtubule structures called mitotic apparatus, which undergoes cycle of assembly and disassembly during the course of mitosis. At metaphase stage of mitosis, it is organized into two parts, a central mitotic spindle and a pair of esters. The spindle is a bilaterally symmetrical bundle located at the opposite end of the cell. An ester is a radially array of microtubules at each pole of the spindle. Formation of the mitotic spindle involves stabilization of the microtubules radiating from the centrosome. These microtubules are of four types: kinetochore microtubules, which are attached to the centromeres of the condensed chromosomes. Centromeres are associated with specific proteins to form kinetochore, where these microtubules are attached. Second type is chromosomal microtubules attached to the ends of chromosomes via chromokinesin. Polar microtubules are not attached to the chromosomes, they overlap with each other and get stabilized. Estral microtubules are the ones which extends outward from the centrosome to cell periphery and have exposed plus ends. Now, the summary of the lecture. Cytoskeleton is a cytoplasmic system of protein fibers which is critically involved in cell motility and cell support. There are three types of fibers: microfilaments, intermediate filaments and microtubules comprise the complete cytoskeleton. They are involved in the movement of cell and its organelle. So, they play an important role in major cellular process, and ATP is also involved in their structural organization. Thank you.